afternoon, everybody, and thanks for the opportunity to talk about my research interest. I am Marisa Roberto, a professor in the Department of Molecular Medicine here at Scripps, where my laboratory studied the mechanism of action of drug of abuse and alcohol in particular. So alcohol or ethanol, I will use this terminology interchangeably, is the most used and abused recreational drug. So the good news is that not everybody that drinks a glass of wine becomes dependent. So let's make sure that I explain what we study in the lab. So social drinking in a subpopulation of people becomes problematic and abusive, and then leads to alcohol dependence in less than 10% of the people. At this point in this progression to dependence, there are significant changes that occur in the brain, in specific brain region, and there is a recruitment of the stress system happening. So there is a shift from drinking to feel good to drinking to feel normal, because at that point, when there is the pathology, people drink to alleviate the anxiety and stress, the malaise that they experience when they don't have the drink or the drug, because these apply to all drug of abuse in general. So there is the excessive and uncontrollable drinking, because individuals lose control over the amount and the times that they drink. And it's a vicious cycle that is progressive. So one factor to consider is that alcohol use disorder, it's a really big health issues throughout the world and accounts for thousands of deaths every year. And if you consider that excessive drinking is comorbid with anxiety, depression, stress disorder, that really makes very hard for individuals to maintain sobriety, especially when it comes to time like this that we are experiencing the pandemic, where there has been a significant increase in sale of alcoholic beverage, and pointed to the fact that people really drink more almost as a coping mechanism with the uncertainty of the current and future situation. And one evidence is that, like alcohol use disorder also increase the severity of COVID-19 symptoms. So I mentioned that in the progression to become dependent on alcohol, there are different changes in the brain that take place. So without going in detail on the complexity of this schematic, where you have brain regions that are color-coded according to the stage of addiction. The amygdala is this red brain region that we study in the lab. And this brain region is really important for the negative effect, for the anxiety and stress component that are characteristic of addiction. So what we call it is really the dark side of addiction, or what for me it's the devil part. So we use a mechanistic approach. So what really we look at is how the communication between neurons is changed with chronic alcohol consumption. So we look at the synaptic changes, and those are electrical recording we do of the activity of this neuron, not only in the amygdala, but also in circuitry that connect the amygdala to the rest of the brain. And we look at specific molecular target. So the overall objective of our research is always to identify the cellular and molecular mechanism in specific brain circuit and neurochemical system that really characterize the disease. Then when we know how, when, and where these changes occur, can we reverse them to prevent the negative emotional state, as well to decrease the excessive drinking that are the phenotype of addiction. Then ultimately, what we wish to do is to evaluate the efficacy of these identified druggable target, not only in the preclinical animal model that we use in the lab, but hopefully in the human laboratory model of alcohol dependence. And I will give you an example shortly. As I said, we take a mechanistic approach, we use in vitro slice preparation of the amygdala. The amygdala drives the negative effect, as I mentioned already. This brain region contains GABA neuron mainly. GABA is the inhibitory signaling, the main inhibitory signaling in the brain. And those neurons in the amygdala also produce the stress peptide, the corticotropin releasing factor that is fundamental in the process of becoming dependent on alcohol. And our lab has shown that CRF not only is the main physiological regulator of the stress response, but plays a key role in dependence. So with this in mind, we take the electrophysiological approach, and my early work wanted to answer two basic questions. Does acute ethanol affect GABA signaling in the amygdala? And does chronic ethanol consumption that induce dependence affect those synapses as well? So we use mice and rats. We make this animal dependent. And what we study is the GABA signaling, whether it's a bulk or spontaneous. And we found that in the dependent animals, so animal treated for weeks with alcohol, have an elevated GABA responses, as you can see here, compared to the non-drinker, the naive animal. Then when we acutely challenge the slices with ethanol, mimicking really what is happening during the acute intoxication, we further found an increase in GABA with ethanol. This is what we see in the slices, but we were also able to measure GABA level in freely moving animal with the dialysate, and most importantly, blocking the CRF1 receptor that are the main receptor that mediate the stress response. The ethanol effect are gone. That really points to how the ethanol and stress system are connected and synergistically increase GABA in the amygdala. So we were also interested in observing if these changes that seems to be very correlating to the dependent state in rodents would be also observed in the non-human primates. So in a collaboration with the Oregon Center, we were able to study non-human primates, and you can see that the amygdala of the drinker display significant elevation in GABA, pointed to a cross-species hallmark of dependence with this compromised GABA. So the approach we have taken in the lab through 2 decades is really to study the cellular mechanism of how this phenomenon is happening. I will spare the detail, but obviously we look intracellular pathway that lead to the GABA release, including calcium channel. And another aspect we are looking at is whether other neurochemicals that are present in the brain are changed with chronic alcohol abuse. And we divide them in generally pro-stressor, so those neurochemicals that actually increase GABA, and the anti-stressor, and how they actually are on the flip side of the coin, trying to decrease GABA, so being the anti-stress. So when we know how these mechanisms work, we take several approaches. One approach is to try to ameliorate these changes. And for instance, several years ago, we tested gabapentin Gabapentin is a FDA-approved drug for the cure of epilepsy and shingle pain. We tested in our in vitro slice, and we found that gabapentin decreased GABA, and most importantly, decreased excessive drinking that is associated with dependence. Those data were really important for a later randomized clinical trial conducted here at Scripps by Dr. Barbara Mason that she demonstrated that gabapentin is actually able to decrease and alleviate drinking in a subpopulation of patients. We were really pleased to see that. Another way we do this, because we know GABA is compromised, elevated with dependence, we take advantage of the amazing collaborator here at Scripps, and particularly the chemistry department. So in a recent work with Ryan Shandy, we were able to test some of his synthetic GABA mimetic compounds. And we did find that some of the compounds Ryan has produced are really effective in regulating GABA. Another approach we take is to study the neuroimmune signaling as an integrator of both alcohol and stress response. A lot of meta-analysis data from post-mortem tissue of alcoholics point to the lifetime alcohol consumption being exponentially correlated to the brain neuroimmune activation. So there is neuroinflammation that develops with chronic alcohol consumption and lead to the cytokine hypothesis of alcohol use disorder. So the way we approach this is to look really at specific pro-inflammatory signaling and anti-inflammatory signaling. So looking at interleukin that I list here a few, and how manipulated those systems can affect drinking. And this is work through the INEA consortium that basically has the main goal to repurpose FDA drugs already approved for anti-inflammatory disease as anti-inflammatory for several diseases, but taken to the alcohol use disorder. So one approach we took, because microglia are the main primary immune cell in the brain, we wanted to ask whether alcohol dependent activate microglia. And yes, it does. Looking at central amygdala and cortical area, there is a significant activation of microglia with chronic alcohol consumption. Then the question was whether depleting, blocking microglia would affect the drinking phenotype. And surely did. We depleted microglia successfully with Plexigon, and we observe a significant decrease in the drinking associated with dependence. And when it comes to GABA signaling, actually depletion of microglia decreased the elevation in GABA that we observe in dependent animals. So to bring it all back together in our schematic, we still have the synapses. Now we have added cytokine and the receptor. We have a different intracellular pathway and mediator of the cytokine effect. And in addition, we also have microglia as a new druggable cell type. So our model, our intent is to test through the INEA consortium anti-inflammatory drugs that are FDA approved, and to assess whether they normalize, they compromise GABA, and they also alleviate the drinking phenotype. With this, I would like to thank you for watching, and I really want to thank all the current members in my lab. Without them, this work would not be possible. Obviously, the former member as well, and my valued collaborator at Scripps and outside Scripps, and the generous funding from NI888 and the Pearson Center. Thanks.
Hi, my name is Sean Joseph. I am the Vice President of Pharmacology at Calibr, a division of Scripps Research. We like to think of ourselves as the drug development arm of Scripps Research. Today I want to talk to you about immuno-modulatory approaches to neurodegeneration. And neuroinflammation is associated with Parkinson's disease as well as other neurodegenerative conditions. And this neuroinflammation results in the presence of activated microglia that contribute to an inflammatory microenvironment within the brain. The pro-inflammatory environment, this pro-inflammatory environment leads to dopaminergic neuronal cell death. Interestingly and importantly, peripheral administration of GMCSF leads to neuroprotection and decreased microgliosis through a number of potential immune-mediated mechanisms. Treatment with GMCSF may result in the generation of tolerogenic dendritic cells that can indirectly induce regulatory T cell populations. It can also direct induction of immunosuppressive regulatory T cell populations themselves. And also they could potentially shift the overall CD4 positive T cell subsets into a more anti-inflammatory phenotype. And importantly, following peripheral immune alterations and cell inductions, regulatory T cells and or anti-inflammatory CD4 positive T cells can cross the blood-brain barrier into sites of inflammation and restore the brain's microenvironment into one that is more neurotrophic and anti-inflammatory, leading to increased neuronal survival and health. And importantly, this is true for Parkinson's disease, but it's true for many other neurodegenerative diseases as well, such as Alzheimer's disease, ALS. It's even been shown to play important roles in MS. And then outside of neurodegenerative diseases, there are other indications for which this mechanism seems to be important, including acute radiation syndrome, autoimmune diseases, lung disease, and Crohn's disease. And so what we've focused on at the moment is Parkinson's disease, but there are obvious, other indications that we're also pursuing that we're not going to necessarily cover today. And so what we found was that, GMCSF is a very interesting target. It's been shown preclinically to modulate that immune or that inflamed microenvironment that I just mentioned. It modulates the innate microglial immunity and then increases regulatory T cells that then make their way from the periphery into the brain, resulting in an anti-inflammatory and neuroprotective phenotype. And this is some of the data preclinically that has demonstrated a very interesting effect of GMCSF treatment. And if you look at the top right, these are many different labs demonstrating preclinically in animal models of Parkinson's disease that GMCSF treatment increases T regulatory cells dose-dependently, and then that then leads to an increased dopaminergic neuron and improves overall locomotor activity in Parkinson's disease models. And importantly, as I mentioned earlier, the effect is not only for Parkinson's disease, but other neurodegenerative diseases such as Alzheimer's disease. GMCSF treatment in animals has reversed cognitive impairment in those animals and reduced amyloid burden in an animal model of Alzheimer's disease. And what really led us to looking at GMCSF as a potential target, and I should have mentioned that essentially what we're doing is taking GMCSF and making it into a longer acting version of GMCSF so that we can overcome many of the inherent limitations of the currently marketed drugs, sargramostim or leucine. And what really drew us to this indication and to this therapeutic as a target is work that was done in Howard Gendelman's lab, in which he in Parkinson's disease patients demonstrated clinical proof of concept for the idea around GMCSF treatment, increasing T regulatory cells and then improving disease in Parkinson's disease patients. Interestingly, a phenotype of Parkinson's disease patients, as well as other diseases such as ALS and Alzheimer's, is that these patients have abnormal T cell phenotypes. And this is true in terms of the number of T regulatory cells that are present in the body, but also the function of them is dysregulated. And this dysregulation correlates very closely with the pathology and the disease severity of these neurodegenerative diseases. And so, as I mentioned, in Howard Gendelman's lab, he was able to demonstrate clinically that leukine or sargramostim was able to increase T regulatory cells and improve motor function in Parkinson's disease patients. And when you talk to Dr. Gendelman about these results, the effects are dramatic. He was talking about patients who were wheelchair bound being able to walk, or patients who weren't able to drive being able to drive or mow the lawn, things that their daily independence dramatically increased as a result of treatment with this clinically available GMCSF. Unfortunately, leukine or sargramostim is inherently limited by the fact that it has a very short half-life, a very rapid clearance. And so the molecule only sticks around in the body for, its half-life is 1 to 3 hours. And so there's a requirement that you need daily subcutaneous injections. And this can be very difficult for a patient with Parkinson's disease, as you can imagine, to self-administer. And so this was an opportunity in our eyes. We saw this as an opportunity to develop a long-acting GMCSF that then you could dose subcutaneously 1 to 2 times per month, and that this would likely increase efficacy, improve compliance, and have relatively fewer side effects as the peptide itself. And as I mentioned, that the neurodegeneration or the neuroinflammation and its impact on neurodegeneration is generalizable in the sense that there's been many results demonstrated in multiple labs showing that even in, say, ALS in this particular context, that T regulatory cells are able to extend stable disease phase and prolong survival in these animal models. This can be done by transferring T regulatory cells, as you see in the top right of this graph, or you can also treat with a therapeutic that upregulates T regulatory cells. In this case, it's IL-2, but obviously GMCSF is something that would also do this, and we're going to demonstrate that in a collaboration with Columbia University. But at this point, the data that exists is shown here, and it demonstrates a significant prolongation of survival upon upregulation of T regulatory cells by a molecule that you're treating the animals with. And this has been also extended into the clinic. In ALS patients, just as in Parkinson's disease patients, these patients have low levels of T regulatory cells, and they have dysfunctional regulatory T cells. And those dysfunctional T regulatory cells correlate very closely with the ALS pathology and disease severity as well. And so those, interestingly, Tregs with that impaired suppressive function, they function normally when the cells were taken out and expanded. And infusions of those expanded T regulatory cells, which had restored function, those, when you put them back into patients, were safe, they were well tolerated, and then the numbers in the suppressive function increased after the infusion. And across a range of disease states within ALS, disease progression dramatically slowed during these infusions of T regulatory cells, but then rapidly increased following cessation, suggesting that continuous production of T regulatory cells is necessary to capitalize on this phenomenon. And hence, again, a significant opportunity exists for a long-acting GMCSF that would be expected to sustainably increase T regulatory cells and hence slow ALS disease progression. And we believe that a long-acting GMCSF that can be dosed subcutaneously 1 or 2 times a month would be preferred versus repeated Treg expansions and reinfusions, which is cumbersome and difficult to scale. Now, how do you make it a long-acting GMCSF? Well, it turns out that an invention, a discovery at Scripps Research enabled this, in the sense that I think it was probably at this point, almost a decade ago. It was found by Scripps researchers. They identified a unique bovine antibody that had this ultra-long CDRH3 region, and on the end of that CDRH3 region is a knob-like domain presented at the end of an antiparallel beta stalk. This really was very interesting. You could replace that knob-like domain with a therapeutic peptide or a therapeutic protein, and then you could get rid of the bovine portion of the antibody and replace it with a human scaffold, one that preferably had already been in the clinic and demonstrated low immunogenicity and tolerability. And then you can optimize that protein and that fusion protein, essentially, and this results in a molecule that retains the wild-type potency of that peptide that you fused in, and then it allows you to have the extended pharmacokinetics that you're looking for, that long-acting characteristic, and hence the persistent pharmacodynamics. In the context of GMCSF, this is obviously T regulatory cell upregulation. And importantly, this fully recombinant molecule has a developability and manufacturing consideration that mirrors monoclonal antibodies. It's as easy to produce as it is to produce a typical monoclonal antibody. So obviously this early discovery at Scripps research really revolutionized how you can make a peptide longer acting, and it inspired the generation of a new class of antibody agonists and antagonists with excellent pharmacology and manufacturability. Now, some of the data around our long-acting GMCSF fusion protein, what we call PDM608, we've fused it into a couple of human backbones, Synagus and Herceptin, and we've looked at others as well, but we found these to be suitable. These are both antibodies that have been in the clinic for years, and so we very much understand what they do in people. And so we spent a lot of time from a protein engineering standpoint optimizing the expression yield, the stability, the low inherent immunogenicity, and essentially tinkering with the protein to make the longest acting version of the peptide that would also be low in terms of its immunogenicity risk, and it would extend that serum half-life. We also reduced the Fc mutations that were incorporated so that we could reduce the ADCC and CDC and overall immunogenicity of the protein. Now, as you can see here, we were able to get a molecule that is a picomolar potent for our antibody fusion, our long-acting GMCSF. And when we put that molecule into animals, it had a half-life of approximately 200 hours, compared to 1 to 3 hours for wild-type GMCSF. Now, we put this molecule into this optimized lead candidate into animal models of Parkinson's disease. And what we saw is that you get a nice dose-dependent increase in T regulatory cells. And importantly, those T regulatory cell numbers were increasing, but that's not all. We also had an improvement in T regulatory function. So we have a molecule now that, compared to the wild-type peptide, is much more potent in inducing the T regulatory numbers, and also improving its function of those T regulatory cells. In the same animal model, we were able to demonstrate that you get a reduction in that inflammatory milieu in the brain, and in this animal model, we're able to reduce the microgliosis that is part of the pathology for this disease, and then we're able to protect overall the dopaminergic neurons. And this is all incredibly important for a molecule to be able to do in order to improve neurodegenerative disease. Now, we went on further to demonstrate that the way that our molecule is working is through essentially shifting the CD4 T cell population into an anti-inflammatory population of cells, or a phenotype. And this is what we think is resulting in the dampening of the inflammation in the brain. Now, really importantly for a long-acting molecule is durability. And we see with a single dose of our molecule, and we've seen this now in rodents, and we've seen it in cynomolgus monkey, that a single dose of GMCSF, of PDM608 specifically, results in an increase of up to 20-fold in T regulatory cells, and increases their function in data that I showed earlier. And importantly, the increase in T regulatory cells lasts for approximately 12 days following a single treatment. And it was well tolerated in non-human primates and mice. Now, next steps for the program is, we're very interested in developing this for Parkinson's disease, for Alzheimer's disease, and for ALS. And so we're working on getting the preclinical data to support the ALS program. And so we worked closely with the core at Columbia and the ALS, Project ALS Therapeutics core at Columbia University, with the world experts in ALS. And they're helping us to design and also execute on these animal models of disease, but also they're helping us to translate that into something clinically meaningful and to a clinical study design, and potentially partner with us to put this molecule into humans in the future. And to give you a little more granular feel for where we are, these are all the development activities that are ongoing in order to result in an IND submission by the end of 2021 this year. And we hope to get into a healthy volunteer population by the beginning beginning of 2022. And we then want to roll right into an ALS and a Parkinson's disease phase 2 clinical trial and waste no time. I want to thank all of you for listening to my presentation and for learning more about our ongoing project, which is very exciting to us. And we also want to thank all of these folks who made this happen. Thank you very much.